Welcome to the Biotech Startups podcast by Exceeder. Join us as we speak with first-time founders, serial entrepreneurs, and experienced investors about the challenges and triumphs of running a biotech startup, from pre-seed to IPO, with your host, John Chi. In our last episode, Jimmy reflected on grad school grit, his move to Silicon Valley, and lessons from Willow Garage during the rise of ROS. He also shared how building the first biology cloud lab at Transcriptic shaped his bias for speed and focus, guided how he hires for mission, and led to later roles at Built Robotics, Robust AI, and Ginkgo before meeting his Monomer co-founder. If you missed it, check out part 2. In part 3, Jimmy talks about founding Monomer, starting with consulting to validate the problem, then honing a thesis around autonomous cell culture, and industrializing in vitro disease models. He also shares how Monomer integrates instruments with a vertically integrated stack, real-world deployments across tissue types and customers, and why their GTM includes software-only landings, pilot runs in Monomer's lab, and full robotic work cells for teams ready to scale.
When you guys came together and and then ended up founding Monomer, what were the early days like?
Yeah, I guess I had a background in biotech and robotics. He had a strong background in software engineering and very passionate about design. And so the first 2 years of Monomer, we were more of a consulting company, which I think is a great way to start a company because you get to know your co-founder, you make some money. We also hired our founding engineer, Carter Allen. So it was 3 of us and got to have experience building a business. We even made it to becoming profitable. And then over time, realizing, coming up with a thesis by getting hands-on experience, helping other biology labs get started.
And how did what was that thesis and how did you refine it?
An alignment of our interests, like I mentioned, really interested in cells, seeing various companies asking us to build automated cell culture work cells. And from there, the thesis got started around building robotics and software to grow different for cell development.
Very cool. And what, when you had that thesis, what, and then you're comparing it to the state of the market, and the status quo, what is the status quo and how does this Monomer's secret sauce disrupt the status quo?
Yeah, we went in thinking about that application, which I think was very fortunate. We went in thinking, how can we industrialize cell culture? And that later involved to how can we industrialize in vitro systems? So in layman's terms, in drug discovery, a lot of drugs are tested before they're tested on human, some animals, often it's in mice. Before tested in mice, it's tested in vitro, meaning disease models in a dish. And so we've taken that on as our mission to not replace, but reduce the need for animal model testing. And so we help scale the growing of stem cell-derived disease models in a dish to drive down reliance on animal models. I don't think it'll completely go away. I think it'll always need to be there, but at least we can do earlier testing at a cheaper way on in vitro systems.
Very cool.
Through working at Stratios, through doing that consulting business at the early days of Monomer, we always saw, for the smaller companies, always a room in the corner that was for cell culture, usually had no window, fluorescent lights.
Yeah.
Biosafety cameras, fans, those are really loud.
Yeah.
Things are buzzing. And that did not look like fun to me.
Yeah. It is not.
And then seeing scientists repeatedly every day going into the lab, taking cells out of the incubator, looking through them in a microscope, looking at the cell morphology to see if they're healthy or unhealthy, and then making some kind of judgment call on that. And then through their experience from looking at the cell morphology, deciding what to do next, whether it's passaging cells or changing media, adding some other growth factors or other reagents, and then going to the biosafety camera, doing the pipetting and putting it back in the incubator. Often taking like half a day every day, Monday through Friday, but also the weekends. And I'm surely like this can be done better. And
That was my job, by the way. Like what you're describing is like I did a lot of.
yeah, I took me back to undergrad. I had to do some of that. That wasn't my only job. So like, yeah, I learned a bit of cell culture.
Yeah.
And then tying that to my robotics background, seeing, people are looking at cells. And then making some kind of decision and then actually doing something in the physical world. Well, that's a very typical robotics loop, and I felt could be done autonomously.
Very cool. Talk about like the technology that you guys have developed to do that.
Yeah, so now we integrate common lab instruments into automated work cells with our cohesive software suite of workflow automation, data analysis, and decision-making tools. So the robotics automates the tedious cell culture that's done manually today a lot. It increases consistency, reduces biological variability. Then our software provides real-time visibility. It lets you look at all your microscopy images, sample health, morphology, growth. And then that scientists can then remotely manage and collaborate on their cell cultures. And then besides robotics and software, our third pillar is biology. We go really deep into understanding the biology to help our customers with protocol development and quickly onboarding new tissues, and also making it easy to optimize new protocols.
Very cool.
Yeah. So to date, we have deployed half a dozen robotic work cells, grown about a dozen different tissue types, and very complex tissue types from like stem cell derived tissues. The big three in like toxicity is usually cardiac, liver, lung, and we've also done brain tissues.
Very cool.
All the way from like single monolayer cells to like 2D to 3D organoids.
Badass. And like, I mean, I can't imagine this was all done like in one go. Like, kind of like walk me through, like you talked about like the three pillars, like the evolution and how you got to where you are, like where Monomer is today, like you and your team.
Yeah. Like I mentioned, the first 1 or 2 years was like consulting. We like helped labs spec out different instruments, help their lab build it out. And as we were doing that, we would offer different software applications to write for them. So Mark wrote a lot of different single purpose like applications that kind of glued things between different softwares. Then over time, we formed this thesis around autonomous cell culture and helped companies automate their cell culture.
And how hard was it to like actually start getting the hardware and the software like to play nice?
Yeah, I like to say robotics is a game of inches. Like it's all these little things have to come together for the fully automated robot to work, and then the biology has to cooperate as well. So there's a lot of attention to little details and a lot of teamwork, and that no person can do it all on their own. I enjoy building multidisciplinary teams, making them talk to each other, and like you are forced to like collaborate and like understand each other.
And did you have to work with the manufacturers of the equipment to make it all play nice, or is it just out of the box? This stuff just can be integrated?
Oh yeah, I think I look at the software as the glue between all these instruments, and try to form good partnerships with different instruments. Um, Taking inspiration from ASML,
Yep.
building world's most complex machine, but they look at themselves as systems integrators, not necessarily making all the parts themselves, but having good understanding of the application then lets you uh, glue all those different things together
Very cool. And
as the value add.
and what did it take to like to get the manufacturers of these equipment excited and on board with
Yeah,
with your guys' integration efforts?
yeah. So a lot of lab automation is the industry is like started with like uh, different automated instruments, like automated liquid handler, automated incubator, automated microscope, and then I look at that as a modular layer that we can then glue together with different software. A lot of uh, software tools are very horizontal. So there are companies that are focused on just scheduling and open loop executing protocols across these different experiments, which I think was fine as the industry was started with like screening. And so, for example, small compound screening. So trying like a million different compounds, searching through a whole library, trying to find the hit on yeah, causing some effect in some disease pathway. That can like be done open loop, like blindly, where you run it for several weeks, and at the end you look at all the data. Then there are other tools like that can help you manage your data, and they don't necessarily talk to each other. And so with Monomer, what we do differently is focus on one particular vertical of cell culture. But then vertical integration, a la Clayton Christensen, like the from those like
Yep.
strategy books I read
Yep.
when I was a teenager. Industries change from being horizontally organized to being vertically integrated. And for cell culture, we thought the vertical integration is key because I saw scientists looking at cells after taking them out of an incubator, making some decision, acting on them, putting back in the incubator. So that loop, I thought, requires a good execution layer and a good data management layer to then enable this decision making.
Absolutely. And I think, the vertical integration, just like, I mean, I think about Exceeder and like, we're always looked kind of like when other leasing companies are like, are compared to us, they're just like, you guys are weird. You guys are only do lab equipment. Like this is a strip, like, but exactly what focus. It's like the focus and like being able to find that like alignment within the vertical. And then like the longer you do it, the deeper those relationships get. And you're also your expertise too, right? And you just get so deeply familiar with the domain.
Yep.
That it unlocks like magical things. Whereas like, we talk about the horizontal, you just don't become, like it's hard to become excellent when you're just like you're spanning so many different domains.
Yep.
And it's impossible to be amazing at everything. It's kind of this thing where you're like, we don't do any leasing for anything outside of the laboratory.
I see.
But that's a sacrifice that we make in order to become domain experts in this vertical. So it's a similar thing, like you're talking about like cell culture. There's like a deep, like making sure there's this like everything sits flush, like versus is kind of like just have this horizontal thing where it's just like, oh, this is okay, not it's like suboptimal.
Yeah.
So I see a parallel in what we do.
Yeah. I think industries shift from vertically integrated to horizontally integrated at different times in history.
Yep.
Makes sense. I often reread Clayton Christensen's Innovator's Dilemma, who goes in depth on disruptive innovation. But yeah, if you look at the computer industry, like vertical integration is needed when you need to create groundbreaking innovations. So like think like the iPhone.
Absolutely. And now that you kind of like you got your thesis. It's like you've polished it, and you have these like the three pillars for Monomer. What is your approach to like the go to market for this? It seems pretty, pretty novel, and I'm going to imagine it takes a different kind of go to market motion.
Yeah. So now we have these three pillars of robotics, software, and biology, focusing on cell culture, but still that's pretty wide. Then cell culture, we decided to go after like where the most pain is. So we found the most like challenging cell culture protocols, and therefore looking at stem cells because they're very finicky. They like to spontaneously differentiate. There's a lot of domain knowledge. Scientists will spend a whole PhD focused on a particular stem cell line, and only they know or have like a green thumb for growing that type of cell, all the way to the point where they even have trouble explaining it to other scientists and scaling. So like,
Yeah.
it's impossible to scale because you're like, as a company, you rely on one scientist to stay at your company.
Yeah. And did you surface that pain via like just conversations? Did you do market research? Like, how did you come to this insight?
Yeah, a lot of it is through like our consulting in the beginning, and doing custom projects. And I learned like ASML, like kind of started the same way, like a lot of custom machines for people.
Yep.
And over time, we kind of see the pattern on what's difficult and what's easy and what are the pain points.
Got it. And so you've targeted the pain. And that's a wise thing to do. It's just like kind of there's another saying, just like, are you a painkiller or you're a vitamin?
Yeah.
Right. You want to be the painkiller.
Yeah. Or are you a solution looking for a problem? Are you a hammer looking for a nail? Or
Yeah.
So
exactly.
we'll obsess over the problem. Is one of our values.
Yeah.
Certainly, no one, I think, within biology loves like doing cell culture on the weekend. So
yeah, for sure. And so you've targeted, you've found that pain, which by the way, like a lot of people never actually find the pain. And they just spend like they're going down roads that are ultimately dead ends. So you found the pain, which is like, important. Now you're like, okay, we need to actually start getting this into the hands of, the users. Are these like, where you're like, okay, these are small companies, big companies, academic, government, or was it just like, all of the above?
Yeah. In the beginning, there's a lot of like startups who like to innovate, early adopters.
Yep.
Let's see. I'll highlight one of our customers, Rosebud, who was a 3-person startup, and we were able to get them growing 1,900 organoids at a time with our robotic work cell, just one operator.
Cool.
So that was a really great win. We brought up that work cell in like 6 weeks. And then since we found other startups and then have worked our way up to bigger companies, we've deployed as big as several 100-person biotech.
Cool.
It's a gene therapy company. And yeah, on our website, you can see our various customers, including Prime Medicine, where the co-founder invented CRISPR prime editing, base editing. So yeah, it was really cool to work with such amazing scientists. And then now we've deployed also at a core facility, and are working with a core facility at a hospital.
Cool.
Yep. So our customers are like cell line development teams within biotech or pharma, also known as stem cell biology teams or cell biology teams that create cell or tissue models that then get used downstream for testing. Other organoid startups we fit really well, and then core facilities.
Very cool. How does like the strategy on getting a deal done differ with all of them? I mean, it's kind of, my wife she works at Cloudflare, and all the sales motions are so different when it comes to small businesses, mediums, mid-market, enterprise. she works in the government, so she's selling to the government, which is a completely different beast in its own. While
Oh,
we're like some like, not to say that they all have to be different. Maybe it is quite like we just like, here's the pain, you're all experiencing the pain, so easy enough, like let me solve the pain for you. Did you have to like customize your approach with each of them, or was it different?
yeah. I say like the hardest thing for us is like the timelines. So the startups really quick. Biotech's a little faster, big pharma long,
Yeah.
a lot of relationship building. And yeah, we're also in the process of the Japanese big pharma and learning a lot of that is like a lot of relationship building too, which I think is also enjoyable.
Yeah.
Uh, yeah, as a startup, you have to think about, cool, is there a way we can get started faster?
Yep.
So we can have the luxury.
Yep. Absolutely.
It's taking very long. And so this year, so before we'd always try to sell fully automated robotic work cells. And then this year we've had 2 deployments now of like software only for manual workflows, where it's still focused on cell culture, but it's protocols that are done manually using, but we collect data from, let's say, a microscope and single cell seeder, and then manage the workflow and the data coming off of instruments, but without a robotic arm.
Got it. And I'm going to imagine a robotic arm, there's like room for the robotic arm to play at. It's kind of like stepwise, kind of like you one step at a time, like crawl, walk, run.
Yeah, we call it a fast land.
Yeah.
And later we can expand to fully automated robotic work cells once we've like started a relationship and like removed some pain points.
Prove some value like in the very beginning. Like totally. Like I think too, it's just like like Cloudflare does the exact same thing of like that, like the freemium model.
I see.
Right. You're just like, all right, here you go. Like here's the basic free version. The free version is like fantastic.
Yeah.
But eventually they're like, oh, I need those paid features like a lot. I now realize it. And sometimes they don't even know the pain, like how painful it is until you get in there and just like show a little bit of value first and demonstrate it and like surface the pain. And then it becomes
the full robotic work cell was a hard sell because our value add is the software.
Yeah.
But the hardware is so expensive. As you know, recent company, I'm sure it's a whole machinery company.
Yeah.
I forget your tagline is wait.
You got it.
Extend your runway, raise it a higher valuation.
Yeah. There we go. Yeah.
Listening to your podcast.
Yeah, there we go.
It's working.
Yeah, it's working. Yes. The subliminal messages. Like, yes.
But now that before the work cell sale was very hard, it required a lot of trust, a lot of upfront costs. Now we have the software land only. So one, it's a lot less upfront costs. And then we also do like, we're a small team, so we can only do a limited set of design partnerships, but we've started lining those up now, which is really nice. Meaning like it's a free trial for like 3 months and then it converts to paid once we've shown the value.
Very cool.
And I'll mention the third option with which you can engage with Monomer. We've now moved into an office and a lab here in San Francisco, where if you're not quite ready yet to buy a whole robotic work cell and have a lot of upfront costs yourself, you can come to our lab and we can run pilot projects. So you bring your cells, you bring your protocols, we'll onboard it, run it in our facility,
cool.
And you get to try it out.
Very cool.
So software only, monitoring your cell cultures, that takes like 3 days to onboard before we can show value. Generating data in our lab with a pilot project, that's maybe 4 weeks to onboard. And then fully automated cell culture after the hardware is all there. It can take as fast as 6 weeks to onboard.
Yeah, I'm imagining like just like, having these kind of like various entry points is important because like sometimes some people are just like, yeah, give me the whole shebang. Like,
happens too. Yeah, that's how we come.
Sometimes that happens. This is like, but you kind of like, it's like kind of the early adopter kind of like curve. And then, there's the kind of the naysayers on the other side. Like you got to kind of like find different kind of wedges into each of those segments because they're all, various people have varying degrees of like conservatism when it comes to adopting new tooling and approaches. So I love that approach.
Yeah, it's meeting where the customer is, but also more reacting to, like, unfortunately today's pretty tough environments in biotech, right? So,
Yeah, absolutely. Yeah, and it's the flexibility. Like it's like, as of right now, the flexibility and efficiency is, paramount. So like I like the way that you guys have adapted on the fly because I think it makes a meaningful impact for your customers, I'm sure. And so, and interesting to go back to the one company, the large Japanese company that you're working with, is like there's like also like different business culture, like when it comes to doing it. And it is very different. Like, I mean, you went to Japan, so you have a, and you live there, so you kind of have a firsthand experience. But like, I guess I'm just like curious, like, what's your experience like? Kind of, now, like, is it you're just like, I kind of know the ropes, like I can kind of get this done.
yeah, to kind of get back to the beginning of this podcast,
Yeah,
my international upbringing, I think, really helped there. One, the obvious, like I had lived in Japan, so I knew
yep,
a little Japanese, so I can like break the ice a bit.
yeah,
Say a couple sentences in Japan, say I
yeah,
lived.
yeah,
But I guess, yeah, from an early age, realizing all the different cultures, right? So like
yep,
Dutch folks, not this stereotype, but they very direct. Japanese people, very much reading between the lines, very polite, and very much care about harmony. And then moving to the US, at least in Philadelphia, people are very what's it called? Not rude, but it's sort of like if people like you, they like relentlessly make fun of you.
yes, I understand that. Yes, yeah, 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 yeah.
That's like a bit of getting used to.
Yeah, my closest friends like rib me a lot, like, so I get it.
Yeah, and it's like a skill set in every different culture. Like being polite is a skill set in Japan. Ruthlessly teasing each other on the East Coast, another skill set.
Yeah,
And like now being on the West Coast, like the teasing doesn't really happen. So I feel I've lost that skill set.
yeah, yeah. But these are real, like these soft skills are real,
I,
like super real. They're like
right,
super real because like, not every single business, and that's why I'm always curious, like, right, like working with the government, working with academia, working with companies that are based in different countries, right? If you're gonna work with them, these like soft things are important,
right.
right? It's not just the technical. Like you've got to appeal to these other aspects that are important to them.
Yep.
No less like we were, one of the partnerships that we were forging was like with also a large Japanese company. When we first got started, I was like much younger, and I'm born in the Bay Area, just like American. I was just like too forward, too quick. Like I was like, why don't we? This just makes sense. Let's get this done now.
Right.
That doesn't fly. Like
Yeah.
that does not fly. We work together now. Just like I'll give you the spoiler now. Like we're partnered now, but it took a lot of, and they were fortunately patient. Like this was their American outpost, so like that I was interfacing with more, so they kind of understood. Like, all right, John, like you're coming in kind of hot right now, but we got to build consensus here. Like back in Tokyo, like just heads up. And just knowing those intricacies and kind of like the soft, I can see like why your international, like how your international like upbringing can really complement this because you kind of you're working with a varied group of folks and stakeholders.
Yeah, and I think the international upbringing always makes me listen first and kind of trying to gauge, all right, what are the norms? What is everyone's style? And I kind of adapt, both in terms of like, yeah, talking to, let's say, a Japanese big pharma versus like a Dutch startup, both of which we have customers. And then But also within like engineering, since we do robotics, which is both hardware and software, and then biology, listening across disciplines.
Yeah.
Like understanding each other, because every problem, since we're vertically focused on specific applications, we can control, do we solve this in software? Do we solve this in biology? Do we use a different releaser that lets the cells come off the well a little faster? Or do we take the plate to a shaker and kind of help and tilt and aspirate, help the cells come off mechanically? And you kind of make all those trade-offs, which I think is super fun.
Super fun, and I think not done enough, because sometimes people are like, you brought up like you're a hammer looking for a nail, like, right? And oftentimes everyone's like domain or specialty is the only way to do it.
Right.
Like you're just like, no, we got to do it, insert whatever specialty it is this way.
Right.
and
that's the best practice in
yeah. But I absolutely agree. You need to cross the line. Like for me, the example of, so I'm a wet lab guy. And obviously, and when I was still in the lab, comp bio was like starting to like get a lot of momentum. But like for us, it's just like wet lab only, like the computer stuff, like not real. Like it's just like, and so there was the rebattling. It's kind of like when we did like X-ray crystallography. It's like we got to, we got for structural bio, like we got to do the lattice, and like we got to like to confirm the structure. And the comp bio people were just like, no, we don't. Like,
no,
we don't.
Yeah, it's hard to talk across boundaries, but every challenge is the opportunity.
Yeah.
I think that's how we differ from a lot of companies in our space. A lot of companies, like I mentioned, like Clayton Christensen, traditionally in our space have been focused as horizontal tools. So a scheduler that can do any protocol, or a data ingester that can ingest any type of data. We slice across those layers and focus on cell culture, specifically the most difficult cell cultures like iPSC-derived tissues, and then get to make trade-offs between all those layers.
And that's where magic happens, obviously. It's like when you can like be cross-disciplinary and collaborative on that front. And it is so funny. It's like, it goes back to when I was thinking about when I mentioned that my dad was like, why can't you do the math and physics? Like just and my dad just like always thinks about everything in the math, physics, like engineering lens.
Right.
So you do it this way. It's like, I don't think like that. Like, I don't know how to explain it. I just don't think like that. But I can like, the problems, like I can, we ultimately come to like a similar place, but just from different angles. Like it just like, there's a bunch of ways you can like problem solve. It doesn't have to be a very specific way. And it's like, it's thinking orthogonally, which is like, I think again, another skill set. It's like understanding soft skills, having those soft skills and using it at the right time. And just knowing who's on the other side, but also like being able to think orthogonally, and just like, and it helps when your co-founders are from different backgrounds. And it sounds like you've managed, again, just like strength, weakness, like we talked about, like finding your limit. Like, okay, this is, I think you said I'm a decent like at software, but I'm not like amazing at software. But your co-founder,
I'm a complete hacker.
Very different. There's like a different, right? Just like,
but I know enough to like make things work and speak the language.
Yeah, exactly. And it's kind of that. It's like that too, right? It's kind of, I think being able to work together across lines and like covering one's weaknesses with someone else's strengths is like a excellent way to do it.
That's all for this episode of the Biotech Startups podcast featuring Jimmy Sastra. Join us next time for part 4, where Jimmy shares why he treats fundraising as just one tool, how Monomer added a services arm and its own SF lab to accelerate learning, and the go-to-market wedges, software only, in-house pilots and full work cells that meet teams where they are. He'll also talk about decisive calls with incomplete information, managing risk in a tough biotech climate, and the vision behind Monomer's AI Scientist Cell Culture Hackathon, plus personal shout-outs and advice to his 21-year-old self. If you're enjoying the podcast, subscribe, leave a review, and share it with a friend. Thanks for listening. The Biotech Startups podcast is produced by Exceeder. Don't want to miss an episode? Search for the Biotech Startups podcast wherever you get your podcasts and click subscribe. Exceeder provides research labs with equipment leases on founder-friendly terms to support paths to exceptional outcomes. To learn more, visit our website www.exceder.com. On behalf of the team here at Exceeder, thanks for listening. The Biotech Startups podcast provides general insights into the life science sector through the experiences of its guests. The use of information on this podcast or materials linked from the podcast is at the user's own risk. The views expressed by the participants are their own and are not the views of Exceeder or sponsors. No reference to any product, service, or company in the podcast is an endorsement by Exceeder or its guests.